Hello, good afternoon, everyone. Okay. Yes, okay. Good afternoon, everyone. Welcome to the second lecture series of Philippine Planktology. Okay, so thank you for joining us again this year. Last year, we did this lecture series, the first lecture series around April to June 2021. So during that time, we invited some of the pillars of Philippine Planktology. Remember, if you joined if you joined those webinars, those talks were from Drs. Willie Campos, Ephraim Mitilio, and Dr. Ray Don Papa. Okay, so this year we are now going international, and so we invited speakers from different countries working on planktology so that we can have some updates on the advances in the fields of in the field of planktology. Okay, so again, thank you for joining us today. This talk or this webinar is brought to you by the University of the Philippines Visayas, particularly the Institute of Marine Fisheries and Oceanology. And also, this is brought to you by the Philippine Society of the Plankton Society of the Philippines, or PSP, and then the administrators of our favorite Facebook page, Philippine Planktology. Okay, so today we will have a talk from Dr. Ryuji Machida, and then in the next lectures, we will have a talk from Dr. Chi Hao Xie and Dr. Shuhei Ban. Okay, so just a few reminders. I think everybody is very familiar with the house rules now on Zoom webinars. So participants are requested to turn off their cameras, but I think this is already automatic now. So also please keep your microphones on mute. If you have questions, please hold your questions until the end of the presentation. And then questions will be entertained during the open forum. And if you want to ask the question, you can raise your hand, and then we can turn on your audio and video if you want to do it live. Otherwise, you can also type in your question in the Q&A box. Okay, so we have a chat box and a Q&A box. Please send your questions to the Q&A box. Okay, and please do not spam our chat box. Okay, at the end of the webinar, we will send the evaluation form link in the chat box, so you can answer the evaluation form, and then this will help you claim your electronic certificate later on. Okay, so I think some participants are trying to practice the hand raising feature of Zoom. Okay, so just a quick look of our program this afternoon. We will have an opening remarks from our director, the info director, and then later on we will have an open forum to be organized by our discussant, Dr. Mark Louis Lopez. And then later on, please hold on, stay for a while because we will have a photo opportunity together with our guest speaker. All right. So at this point of the program, may I welcome Dr. Sheila May Santander de Leon, the info director, to give us an opening remarks.
Thank you, Christy. Welcome, everyone, to the second Philippine Planktology Lecture Series. And this is the 7th session of Historia Lawud, also Stories of Our Oceans. So the Institute of Marine Fisheries and Oceanology, as partner for this event, is very grateful to the Philippine Planktology and Plankton Society of the Philippines for their continuous efforts to bring to our students and stakeholders relevant and latest knowledge on planktology. Of course, our info faculty here and the Vice President of the Plankton Society of the Philippines, Dr. Christy Acabado, together with the members of these organizations, have taken so much effort to put this together and bring this lecture series on its second year already. So we are also honored to have our speaker, Dr. Ryuji Machida, for today's session. Domo arigatou gozaimasu, Machida Sensei. And yes, and one of the aims of Historia Lawud is to strengthen our academic and research collaboration. So I'd like to take this opportunity to announce that our institute is offering graduate programs such as MS Fisheries, major on Fisheries Biology, and MS Ocean Sciences. So the application for academic year 2022 and 23 is now open. We also have other graduate programs under our college, but the details of this will be posted at the end of the webinar. Again, welcome at magandang hapon sa lahat.
Thank you very much to our director, Sheila Santander de Leon. Okay, so she also studied in Japan, so she knows how to speak some Japanese. Okay, to continue, I would like, it is an honor to introduce to you our speaker this afternoon. We have Dr. Ryuji Machida. He is an Associate Research Fellow from the Biodiversity Research Center in Academia Sinica, Taiwan. He received his PhD from the University of Tokyo in Japan, and following that, he did some postdoctoral fellowship also in the same university in Tokyo, and then in the Smithsonian Museum in the US. Dr. Machida is a marine biologist studying zooplankton communities. He utilizes different DNA and RNA-based molecular techniques to study zooplankton ecology, evolution, and diversity. He is also interested in curating DNA sequence reference database, which can be used as mitochondrial sequence taxonomic assignment reference, which he calls the Midori tool. Okay, so I hope you will learn a lot today, and I hope that you will ask several questions to Dr. Ryuji Machida. Please give our speaker a warm welcome, Dr. Ryuji.
Okay, thank you very much for this opportunity, Christy, Louis, also Dr. Director Sheila. I really appreciate this opportunity that sharing that some of our recent studies. So I will share my screen. Oops. Okay. Okay. Oops. Okay, so I hope you guys can see my screen now. Okay?
Yes, we can.
Okay, so today's topic title is that the importance of analyzing RNA in zooplankton ecology. So kind of important keyword is RNA. So today I'm going to talk about basically three topics. First one is that using metatranscriptomic to estimate the diversity and the composition of the zooplankton community. And second one is the development of transcriptomic-based growth rate indices in two model eukaryote and relevance to metatranscriptomic datasets. And then third one is that allometric scaling of RNA abundance from gene to community. Actually, all of these projects have been done by my PhD students. So that the first one and the third one is actually Louis. Maybe I will change the pointer. So first one and the third project have been carried out by Louis. And then second topic is carried out by my another PhD student, Daniel. Both of them, actually Louis already left my laboratory, but Daniel is still continuing my lab. That both of them joined the Taiwan International Graduate Program. So if some student interested, that they can visit our website. Okay, so first topic I want to talk about is that I kind of cannot see. So first topic is that the use of metatranscriptomics to estimate diversity and composition of zooplankton community. So what we have done is that we basically created a mock community using that freshwater zooplankton sample collected Feiche reservoir, and then we sequence those samples. So what is mock community? Mock community is that actually in the tube, we put known number of the several species. So, and then we extract DNA and RNA from those tube mock community and then sequence. And then those samples we collected at the Feiche reservoir. Feiche reservoir is that located in the northern part of Taiwan. And then this is the most important water reservoir for the Taipei area. Okay, so why we have done this mock community studies? Motivation is that we are wondering how the metabarcoding data will be reliable. Basically, we had two concerns. First one is that the PCR amplification bias. You know that in metabarcoding, people amplify a target gene using PCR. But if PCR primer does not match to that some specific taxon, we can easily imagine those species will not be amplified. So this was my first concern. And then second one is that pseudogene. Actually, we already know there is so many pseudogene. Pseudogene is that a non-real sequence copied into the different location of the genome or different location the other genome. So those pseudogene might be amplified during the PCR too. So to make sure if these are happening in metabarcoding data, we carried out this mock community study. So in this study that we create that, hold on, I cannot, sorry. I'm a little bit having difficulty. Okay. Oops. I will continue. Sorry. So in this study, we basically created 5 mock community. Like first one is that the cladoceran dominated mock community. This means that we put 5 individual of that Mongodioptomys copepod, 5 individual of the Mesocyclops copepod, and then also 20 individual of the Bosmina, 7 individual of the cladocerans into the single tube, and then homogenize, and then extract DNA and RNA. So in the second community, copepod dominated mock community, we put more copepods. And then third mock community, we put basically equal biomass of each species. And then fourth community, natural assembly. That means that the species composition resembles the actual zooplankton community in the Fairchild reservoir. And then last one is that with a rare species, that means only one individual from small species put in tube. So we want to make sure if we can detect this small species or not. And then from each mock community, copypod dominant, cladocylon dominant, equal biomass, natural assembly with a rare species, we extracted both RNA and the genomic DNA. And then from RNA, we performed that metatranscriptomic. That means that without amplifying target genes, we sequence everything. And then second is that CO1 amplification using cDNA as a template. And then in case of DNA, after extracting DNA, we amplify CO1. So this is basically the standard DNA barcoding procedure. Actually, this morphology and the metatranscriptomic, to get these data, we are not using PCR. So these two, we consider PCR-free methods. In contrast, these two methods, CO1 amplification from cDNA and the genomic DNA, we use the PCR. So by comparing these two data sets, we can see the PCR bias. And then next, one data set is that the CO1 amplifier from cDNA. Actually, pseudogene will be not transcribed to the messenger RNA. So this data set does not contain pseudogene. In contrast, that this data set using genomic DNA as a template, that means that the sequence may contain the pseudogene. So comparison of these data sets will give us an idea of that pseudogene contamination. Okay, so first result. So this is figure A is that the first result of the species composition. And species composition based on the sequence data set. In case of morphology, this is based on dry weight percentage. And then we also get data from cDNA amplicon, genomic DNA amplicon, also metatranscriptomic. So first result we can observe is that the cDNA, genomic DNA methods resemble each other. Whatever the mod community, cladocylon dominated, copypod dominated, this is, sorry, I cannot say, equal biomass, natural assembly with rare species, all of the mock community, that the PCR, both of the PCR methods are very similar. And then next, morphology and the metatranscriptomic resemble each other. For example, like cladocylon dominated, and then this is result from metatranscriptomic. This is result from morphology. Both of the data pattern is very similar. This similar pattern has been observed from all mock community. And then another thing we have noticed is that the Mongo diaptomus, this is the blue color copepod. Actually, this copepod is most abundant zooplankton in the Fairchild reservoir. However, any of the PCR-based methods, we could not detect this species. So that we could notice that because of PCR bias, that we could not detect the most abundant species in the data set. And then I want to move on to the figure B. So this figure B is basically same data set, but transformed into the 2-dimension. So how we see this figure is that, for example, this black color is result of the genomic DNA. And then this yellow color is the result of the cDNA amplicon. So we can see both PCR methods are very similar. In contrast, metatranscriptomic is that these blue color are very similar to the morphology, which is red color. Another thing we notice is that actually, if their position are located in very close. So that means that the species composition, estimated species composition is very similar. And then since both of the PCR-based methods, the area they share is very small compared to the area that the metatranscriptomic and then morphology shared. So that means that the species composition of the all mock community are very similar, whatever the mock community they used. In contrast, that the metatranscriptomic area is much wider. So that this means that the metatranscriptomic has a much wider sensitivity, much higher sensitivity compared to the PCR method. So this is the first result we observed. And then second, I will talk about the bias of the pseudogene. Basically, we compare the result from genomic DNA, which likely contain the pseudogene, and then cDNA, which likely not contain the pseudogene. So that the first, I want to talk about the ASV. ASV is like a haplotype, basically that sequence type. So in case of that mesocyclops, we observed more than 900 haplotype from this species when we amplify a target gene from the genomic DNA. In contrast, if we amplify a target gene from cDNA, we obtain only 91, so roughly 10 times difference. And then another example is that the number of indels. Indels means insertion and deletions. We sometimes observe the indels from the data which amplifies from genomic DNA. However, in contrast, we didn't observe any indels if we amplify cDNA. So it is this is the result. Very likely this is because of the pseudogene amplified in genomic DNA, but not in the cDNA. Okay, so first conclusion is that the PCR skews the species composition in metabolic coding data. And then some of the species might not be detected if you use the PCR. And sensitivity higher in metatranscriptomic. And then pseudogene are a lot in genomic DNA. So our recommendation for the zooplankton study is that not metabarcoding. We should use the metatranscriptomic. Okay, so next topic is that development of transcriptomic-based growth rate indices in two model eukaryote and its relevance to the metatranscriptome dataset. This is my another PhD student, Daniel's project. Actually, growth rate is a very important quantity in the zooplankton ecology because this is a quantity which can be transported to the higher trophic level. However, estimation of zooplankton is still very difficult. So at the moment, we have roughly two ways of estimating the zooplankton growth rate. First one is the continuous observation of specific individual or the population. For example, this dinosaur, if we can continue observe same individual, and then every year, if we can measure how tall they are, we can estimate the growth. However, that tracing single individual, same specific individual of the zooplankton in ocean or the lake is practically impossible. So that this method is really difficult to use. And then second, biochemical and radiochemical method. Famous method is RNA DNA ratio. However, we already know that this method is not really useful. Actually, the idea that the theory behind this ratio is reasonable. However, that already 1986, that the Conover and the Poulet already mentioned that we must go back to the laboratory under careful controlled condition in developing and calibrating these techniques. So we know that this method doesn't work well. So still that the estimation of the growth rate is very difficult. So now we are proposing that new method, new nuclear and mitochondrial messenger RNA ratio as a novel growth index. So first, I'll talk about the idea of the nuclear and mitochondrial messenger RNA ratio. So messenger RNA is that, as you know, that it's a template for the amino acid sequence. So they can synthesize the protein. And then actually messenger RNA, we can classify into the two classes. One is that those messenger originally encoded in the nuclear genome. And then another is that the messenger RNA which encoded on the mitochondrial genome. As you know that the mitochondria is actually responsible for the making of ATP. ATP is basically the energy currency in the cell. So that all of the energy production, basically all of the energy production in the cell is responsible. And then we can measure from the mitochondria messenger RNA transcript. In contrast, all genes required for growth, such as the cell growth and then cell division, actually all of them are encoded in nuclear genome. So we came up with the hypothesis. If the growth rate of individual is high, we expect that the ratio nuclear and mitochondrial messenger RNA also should be high. In contrast, if the growth rate is low, we also expect that the ratio of the nuclear mitochondrial messenger RNA should be low. So that was the first hypothesis we came up. And then to prove this idea, what we have done is that the culturing of the Daphnia magna. Daphnia magna was cultured individually, and then they were fed phytoplankton every day. And then using the light condition, 16-hour light and then 8-hour dark condition. And then for this experiment, we use that 9 kind of treatment to control the growth rate of this Daphnia. Basically, that the three range of the temperature and then 3 range of the food concentration. And then each condition, we culture the 5 individual. So we expect high temperature and then high food condition that are very likely individual will grow faster. And then low temperature, low food concentration, individual will grow low. After the culturing, we extracted the total RNA. And then from total RNA, we extracted messenger RNA. From messenger RNA, we created the Illumina sequence library and then performed the Illumina MySeq 150 single gene reads. And then after the read is available, we map those sequences to nuclear genome and the mitochondrial genome. Since whole genome of the Daphnia magna is already available, so this process can be done much easier. Okay, so in this study, we are proposing basically 4 new ratios. First one is that the total nuclear and mitochondrial messenger RNA ratio. And second is a nuclear and mitochondrial ribosomal protein messenger RNA ratio. Third one is that the Go term and the mitochondrial messenger RNA. Fourth one is the nuclear and mitochondrial specific gene messenger RNA ratio. Actually, that the Daphnia genome has a not well annotated genome, so that applying this idea that the Go term was not really feasible, so we didn't do in this study. Okay, so the first one is that the total nuclear and mitochondrial messenger RNA ratio. So this is the result. So actually that the correlation coefficient was not so high, but still the significant correlation we observed. And then next one, nuclear and mitochondrial ribosomal protein messenger RNA. So next one, this one correlation coefficient was much higher. And then the, again, result was significant. And then last one is that the nuclear and mitochondrial specific gene messenger RNA ratio. In the preliminary analysis, actually we've tested many mitochondrial genes, and then always that the CO1 did good job. So we choose, in the rest of the study, we always choose the mitochondrial CO1 as the denominator. And then in this example, we are showing that the 3 nuclear gene markers, AR and ETHE1 and OTSA. So all of the ratios, they give pretty good correlation coefficient. So this is the second conclusion. We created the 4 growth rate indices, which can be estimated from the transcriptomic and metatranscriptomic data. Okay, so I'll move to the last topic, allometric scaling of the RNA abundance from gene to communities. This is Louis' second project. So again, so motivation of this project was that what is the meaning of sequence read number? In many metabarcoding study, people usually assume it's a relative, those read number reflect relative biomass. For example, if we homogenize 100 individuals of species A, which has a biomass of 1, and then 10 individuals of species B, which has a biomass of 10, and then 1 individual of species C, which has biomass of 100 grams. If we homogenize all of these species, so equal amount of the biomass, 100 grams, people usually expect we get same amount of the sequence from each species. However, actually I will show the result, but this is not right answer, quite different, because people forgetting about the effect of size. Okay, so what is the size? What is the meaning of the effect of size? Actually, there is already so many studies have been done that the metabolic rate is higher in a small organism. This is the image. You know, x-axis is the biomass, y-axis is the mass-specific metabolic rate. So that in case of mass-specific term, metabolic elephant has a much lower metabolic rate compared to the mouse. This pattern has been known for so many parameters, such as heart rate, growth rate, lifespan. You know, heart rate of the elephant is way much slower than mouse. Growth rate is much slower in elephant than mouse. Lifespan is much longer in elephant than mouse. So that our question is, how about that messenger RNA abundance? Abundance of messenger RNA in unit of mass in elephant and then mouse. Relationship of those were our question. So to answer this question, we have created another mock community, but in this case, we use the model organism mock community. We use the C. elegans, Daphnia magna, fire ant, Drosophila melanogaster, and then zebrafish. Important thing why we use such a model organism is that because size range, C. elegans is about 1 microgram, zebrafish is about 400,000 microgram. So the size range is very large. So we can confirm that this relationship much clearer. Another thing is that all of these organisms, we have a genome sequence already. So the analysis of the obtained data is much easier. Okay, so this is the result. So x-axis is biomass. Y-axis is basically lead abundance in both log transformed. And then this is the data from the C. elegans. And then this is Daphnia. This is fire ant. This is Drosophila melanogaster. And then this is the zebrafish. So as we expected that the abundance of the messenger RNA is very much higher in C. elegans if we think about the per unit of the mass. So we will come back to the question. This, people thought about that if we homogenize same biomass, people expect we will see that the same amount of the sequence. However, based on our experiment, result is like this: 10 times smaller organisms, we likely obtain only half of the sequence. This is because of the size effect. So this was the message from the result we can deliver. But this question still persists. So what is the meaning of sequence read number? And then answer to this question, our answer is that this is the amount of energy they produce. And then those are size effect collected already. If you think about like a mitochondrial transcript, mitochondrial transcript is responsible for energy production, so that ATP production. So this is the energy currency of the cell. And then this quantity is exactly that quantity each different species generate in the community. So this is the message, clear message we can deliver from that our experiment. Okay, so that we want to summarize. So first, we use that. First is that using metatranscriptomics to estimate the diversity and the composition of zooplankton community. We demonstrated that there is a PCR bias in metabarcoding method. So we recommend using metatranscriptomic. And then second, development of transcriptomic growth rate. And then we developed basically the 4 growth rate indexes. And then last one is that allometric scaling of RNA abundance. So that the read number is that relative to the generation of the energy. That's our message. And then the final message I want to deliver is actually you can do only if you extracted RNA. We could achieve this result because of we worked on RNA. Many people are afraid of RNA, but nowadays, all of my students, majority of my students working on RNA. So I want to stress that the RNA is not scary. If you properly handle RNA, actually, this will give us very important ecological information. So this is the message I want to deliver today. And then acknowledgement, final slide. So that Frank and then Zach, he's going to talk about next week. So we are getting help from them. So that's a finish of my talk. Okay, thank you very much.
谢谢你, Dr. Machida.
谢谢.
Okay, okay, yeah. Okay, so now at this point, we will move forward to the question and answer. Thank you very much for your talk, Dr. Machida. I believe that our audience has learned a lot from your talk. But before we go on to the question and answer, I would like to say that we are happy to have over 250 participants now in Zoom. And then we have 160 views on FB Live. So you are very famous now, Dr. Machida. Okay. And then I just want to say hello to our participants from all over the Philippines, from Luzon, Visayas, and Mindanao. We also have some participants from Malaysia, New Zealand, and of course, Taiwan. And then from organizations in the Philippines, such as the Philippine Coast Guard, the BFAR, Bureau of Fisheries, and then also from the Department of Education, Philippines. Okay, so now we can move. I will turn you over to Dr. Mark Louis Lopez to be our discussant for this webinar.
Thank you, Dr. Machida, for that wonderful talk. So during your talk, we collected questions from Facebook live streaming, and then from the participants or from the audience, they typed some questions in the chat box. So I think the first question that I'd like to give is from one of our audience is that, why do you think scientists prefer using DNA, specifically DNA metabarcoding, instead of using RNA? So
Okay. I think this is more like a historical reason. First, whatever the field of the biology, first we start using DNA and then start to analyze DNA and try to answer evolutionary question. Actually, myself too, when I was PhD, I was analyzing DNA and then try to analyze, answer that evolutionary question. But because of this community-based analysis, I think so that was the reason that, people started from DNA. That's why people are still using DNA for the metabarcoding. But I want to encourage people to shift to RNA because that I want to recently this idea coming up to myself that to address that evolutionary question, actually DNA is better than RNA. But if we address ecological question, many cases that RNA is better. So that now I think this is the shift currently happening. So that, sorry, I'm kind of talking too much, but the quick answer is that just because historically we have been using DNA from long time ago.
okay, I see. And then I'm trying to arrange questions based on the outline of your presentation. So another question related to the first question, it's from an undergrad student. Like, he's asking why or what are the things that we need to consider when processing RNA? Because in your last slide, it says, do not be afraid with RNA.
Okay. RNA are actually, I can do 1 more hour talk for this purpose, but basically most difficult part is that enzyme to digest RNA, which is that the RNase is very difficult to inactivate. In case of DNA, there is also similar enzyme to digest DNA, which is the DNase. Actually, inactivation of DNase is very easy. However, inactivation RNase is very difficult. So that one thing, and then, those RNase is actually everywhere, in our hand, basically covered by RNase. But so the RNA experiment, definitely you need to be careful. You always need to wear the glove. So, but if you do those careful processing, that you can get good data. And then detail of those handling, actually nowadays we can find in the many YouTube or the website. So that maybe here I don't talk too much, but so one thing important keep in your mind is basically try to reduce the contamination of the RNA into the sample. So that's a short message. Yes.
And then another more general question, Dr. Machida, is that for some of the audience from, I think from the Philippine Genome Center, he is interested in knowing, aside from your lab, are there any other lab that uses RNA for community ecology? And then his second question is that, do you think the price will become cheaper in the future?
Okay. At the moment, RNA, zooplankton RNA metatranscriptomic is very rare. That's my concern. Still, many countries using genomic DNA. So actually, I really want to encourage to start the RNA analysis. If you follow the other people's study, it's not competitive. I'm really happy to help. And then with RNA, I hope I deliver the good message that if we properly handle the sample, we can get very high quality, high sensitivity, and meaningful data for zooplankton ecology. So quick answer is that still very few. Basically, zooplankton community, I don't know any other laboratory. And then, so price, actually, in case of the zooplankton study, and then using next generation sequencing, most expensive part is either sequencing, so next generation sequencing, so that either working on DNA or RNA, I think doesn't matter much. Maybe in the beginning, you need to buy a new kit for RNA extraction. Actually, that costs some, I should agree. But still, that higher proportion of the money you need for the analysis is next generation sequencing. So that I think in terms of the price, I think the DNA-based, RNA-based doesn't much big difference. Although initial investment for RNA facility is required. Okay.
Okay. And then I think I'll move with the questions related to the first part of your talk. The first question is that, are there disadvantages in using mock communities?
Mock community. Actually, mock community is that when the method is still in the development phase, if people are not sure those method is really work or not, it's like a positive control. That I did with metabarcoding and metatranscriptomics. But at the same time, many people are starting doing that eDNA and then eRNA, and then try to correlate with, for example, visual observation or the catch of the fishes. So that's basically try to correlate that actual organism abundance and then sequence read number. So that advantage, short message is actually this is the positive control of the method, kind of validation of the method.
Thank you, Dr. Machida. Another question related to the first part is that, are there any constraint or disadvantages in using metatranscriptomics in zooplankton diversity estimation? And then how can you solve these disadvantages?
Okay. Personally, I don't see much disadvantage. Some people say that sequence depths need to be much deeper than meta-barcoding. But actually, we are not really experiencing those disadvantage.
Mhm.
And then disadvantage. Maybe one disadvantage, if somebody shifting from genomic DNA to RNA, is that, as I said before, that initially, that the lab need to shift from DNA to RNA. Actually, in my lab, centrifuge, thermal cycler, PCR machine, or the pipette, all of them, actually, I created a small room, which is only used for RNA experiment. So that initially, if the lab want to start the RNA experiment, initial some money might be required. So that
Mhm.
might be the disadvantage.
Okay. And then some of the bioinformatics people from the
Mm.
audience,
Mm.
they are interested on like what is the difference between bioinformatic analysis of metatranscriptome and meta barcoding or metagenomic approaches? Like, is it easier to do metatranscriptomic bioinformatic workflow than the meta barcoding?
Okay, very good question. And actually, meta-barcoding data is much easier to process. But so informatics, we are spending quite a lot of money to establish the protocol too. And then we actually, in the beginning, that Christy introduced me that I'm creating the mitochondrial reference database. Actually, using of that mitochondrial reference database, we can separate the nuclear and mitochondrial transcript. And then whole procedure is still very not, it's getting more and more easier, but I should admit the still little bit difficult than standard DNA meta barcoding study.
Mhm.
Actually, more important now is that the creation of the reference of your target species, like from Philippine oceans. I think DNA sequence from their species is not many deposited in GenBank. Those cases identify species of species based on, for example, CO1 sequence will be very difficult. So pipeline is important. Same time, reference sequence is important.
Mhm.
I believe that many scientists, students can identify species of that Philippine coastal or the open ocean species, zooplankton. Those cases, I really encourage doing that, not DNA barcoding, but second generation barcoding. That means that transcriptomic of those species. If we have those data, that the processing of the sequence from metatranscriptomic will be very easier.
Mhm.
So that's my answer.
Okay. And then, Dr. Machida, there are two questions related to the second part of your presentation. The first question is that why do you think, based on your results, why do you think the nuclear and mitochondrial mRNA ratio is proportional or correlated with the growth rate? I think they're asking behind what's the theoretical basis for the correlation between the nuclear mitochondrial ratio and then the growth rate, why they become correlated.
Okay. First, I will talk about mitochondria transcript abundance. Actually, mitochondria is responsible for ATP production. That means that energy production. So, but at the same time, by creating energy, organism cannot grow. Growth of the organism needs nuclear gene transcript. So that the growth is basically the growth of the cell, and then also dividing the cells. So genes responsible for growing of cell, dividing of cell, actually all encoded on the nuclear genome. So if those nuclear genome transcript if the animal is start growing, that means that more cells are becoming bigger, more cells are dividing to the doublings, so that the gene responsible for cell growth and then cell division is indication of the animal growth. So ratio of the nuclear and mitochondrial messenger RNA will proportional to growth. This is because so nuclear is responsible for the gene which responsible for growth. And then mitochondria is only that energy. That's why those ratio will be the proportional to the growth.
I see. And then the second question, Dr. Machida, is that we have audience majoring in undergraduate students majoring in molecular biology, and they noticed specific pattern in the growth rate indices. So one of the audience mentioned that the correlation for ribosomal RNA ratio, it's better as compared to the correlation of the messenger RNA ratio with growth rate. Why do you think the ribosomal RNA are performing better?
Very good observation. Impressive. To be honest, I don't have a good answer for this question. I have been wondering too.
I see. And then I think I'll go with questions related to the third part. One direct question is that what is the application of messenger RNA scaling in zooplankton ecology studies? Like what they're asking, what's the bigger application of mRNA scaling?
Okay. Okay, I think simple answer is that how we interpret the metatranscriptomic data. That's the most important message. Because, and then, so those quantity is directly measuring the energy component, which is already effect of size collected. And then, that in ecosystem study, the two quantity we want to understand, the flow of energy and material. That the flow of energy. Energy is the quantity of the mitochondrial transcript. And then material is quantity of growth, which I talked about in the second chapter. So that those two quantity is very important quantity in the ecological context. And then third chapter that the scaling is demonstrating that those messenger RNA abundance is already collected quantity, size effect collected quantity of the energy component. So that will be my answer.
I see. Thank you, Dr. Machida. I missed an interesting question from the audience. It's related to the second part. he is asking, is it important to consider gene number copies when doing the ratio calculation?
Okay. Actually, in case of RNA, gene copy number is not important. Gene copy number is that how many copies in the nuclear genome, which is DNA.
Mhm.
But what we are analyzing in RNA abundance, so that gene copy number, actually reaction speed might be different, but in general, my answer is that gene copy number is that only we need to consider if we analyze DNA. That's my answer.
Mhm. And I think we're down to last two questions. And these questions are more personal because we have audiences who are undergraduate students and studying fisheries and also interested to go to graduate school, I think. So their question is that what motivated you to study zooplankton?
Okay. Zooplankton. To be honest, my interest in the beginning was that evolution of to be honest, any organism. To be honest, plankton was not really important, but I liked that studying of the evolution of the animal. Somehow, I'm studying the ecology of zooplankton now, but initial of the motivation was, to be honest, that it doesn't have to be the plankton. And then, at the moment, I'm kind of doing something different, but I still like it.
Mhm. And then I think another personal question from the audience is that based on your presentation, they are thinking that most people use DNA and then only few people use RNA. They want to know what motivated you to use or explore RNA instead of DNA, despite a lot of people using DNA.
Okay. this is because I noticed that the pseudogene in quite early phase of my PhD study. And then, actually, not only me, many zooplankton people who study mitochondrial gene, actually, many people are noticing that there is a lot pseudogene. And then to make sure pseudogene, I need to work on RNA. And then also that cloning of sequence. And then, that time I already learned a new technique analyzing RNA at the time of my PhD program. So that time I was not scared of the RNA anymore. And then same time, I get very good sequence from that RNA. So, and then actually, it's not many people realize, but I'm the first one who did that metabarcoding study. And then that time, I'm already using RNA as a starting template. That time, I couldn't use next generation, but next. So after my paper, people start to use next generation sequencing with that genomic DNA as template. So my impression is that so at the time of PhD, I already used RNA, so I was not afraid. I already had the confidence using RNA. So that I think that is my answer.
Okay. I think I'll end with this last question. It's not totally related with your presentation, but some of the master students from the Philippines, they are asking like, what do you think is the advantage of doing their PhD in Taiwan? I think they're interested to the international graduate program. What do you think
Okay.
are the advantages?
Advantage.
Not sure.
Yeah, for Filipino students, kind of easy to see is that very close. Nowadays traveling different countries is difficult, but gradually, getting easy to travel. And then when we travel, between Philippines and Taiwan is so easy. Actually, people know that I'm Japanese, and then for me, going back to Japan is so easy. I was doing my postdoc in United States, but that time, traveling that States and Japan was so difficult.
Mhm.
But now, and then Taiwan, actually, the advantage of that our program is that we have very good access to next generation sequencing, and then practically we provide a good amount of the stipend too. And then we have a good access to pretty good machines in general. So I think that will be a advantage.
Okay. And then I think, Christy, do we still have time for some questions?
Okay.
If
I'm
there are more
checking.
questions, I think we can entertain one more,
Okay, one more.
or you can already provide your synthesis of the topic.
Mhm. I think I can provide the synthesis. So I think based on Dr. Machida's presentation, the message is very clear. So the general message is the advantages of using RNA over DNA because of because of its higher sensitivity, and at the same time it avoids some methodological biases, such as based on his presentation, it can avoid PCR biases, and at the same time it can avoid the contamination of pseudogenes. Other application of RNA in zooplankton community ecology studies would be its ability to represent or serve as proxy for some physiological processes such as growth rate. So based on his presentation, Daniel, his PhD student, was able to produce or to develop a novel RNA-based growth rate index that can serve as proxy for estimating growth rate, which is very hard to do it in natural environment. And using this index, you can easily have an idea on how individuals, community, or population of organisms are interested are doing well or doing when it comes to growth in natural environment. And then a last part of his presentation, it provides us better understanding on how we can interpret RNA sequence data with regards to its relationship in the size of the organism in the community. This is very important because most of the time, if you are processing community samples, most of the individuals are highly varying in terms of size. So with this new interpretation in mRNA read number, you can easily normalize the data based on the size of the individuals present. So I think it gives us future direction on what to avoid and what can we take note while processing zooplankton community samples. Most of the people are still convinced that DNA is still one of the good standard for analyzing zooplankton community, but slowly the entire community is transitioning to the use of RNA and appreciating its possible application in zooplankton community studies. So I think that's the general synthesis and the message that Dr. Machida wants to tell to most audience. And at the same time, I think it's very important to highlight the possible options of doing your PhD or graduate studies in Taiwan. Both Christy and I are products of the Taiwan International Graduate Programs, and then we were able to finish our degree, I think, in a reasonable time. So why not consider going to Taiwan for your PhD? Yeah, so I think that's the synthesis, Christy.
Thank you
Maybe
very much.
can
Yes.
I say?
Yes,
Okay.
please.
So that today's work is I didn't basically I just gave the order to the Louis and Daniel. So today's result is all work from that Louis and Daniel. That's I kind of want to stress. Thank you.
I think Daniel is also in the audience and is listening.
Okay,
so,
okay.
Hi, Daniel.
Hello, Daniel, and hello everyone in Sinica today. Thank you very much, Dr. Machida and
Okay.
Dr. Mark Louis Lopez for being our discussant today. Again, I would like to say that this webinar is brought to us by the Institute of Marine Fisheries and Oceanology, the Plankton Society of the Philippines, and of course, the Philippine Planktology administrators on Facebook. And also, this webinar is now officially part of the Historia Lawood webinar series of Info in UPV. Okay, so thank you again. At this point, we can now proceed with the awarding of certificate to our guest speaker, Dr. Ryuji Machida, in recognition of his outstanding contribution as keynote speaker of presentation entitled Importance of Analyzing RNA in Zooplankton Ecology at the second lecture series on Philippine Planktology on June 30, 2022, signed by the Director of Info, the Vice President of the PSP, and the Administrator of Philippine Planktology. Thank you so
Okay.
much for doing this for us, Dr. Machida. If you have some last words.
Yes, it's really honor for me too. That also I really enjoy that sharing my recent study. So I appreciate this opportunity. Thank you very much.
Thank you very much. Now, can we probably have a photo opportunity? I am not sure if we can invite the attendees, but Sir Juni, please turn on all cameras if it's possible so we can have a photo. I will stop sharing first. Okay. Hello, Daniel. He's on the chat box. Okay, so let us know if it's done.
So I think the participants can now open their camera or video so that they
Sir Juni,
Okay.
can you open?
I think we must promote the attendees first.
It will take some time, right? So I think
Yes,
the
ma'am.
panelist is okay. Yeah.
Yes,
Okay.
panelist.
Just the panelist.
Okay.
Thank you.
So,
So I'll take the screenshot now.
yes.
1, 2, 3, smile. Another one. 1, 2, 3, smile. Okay.
Okay. Thank you so much. Let me go back. Okay. So let me go back here for a while. Sorry. Okay, so right now I would like to take this opportunity to introduce to you the programs offered by the Institute of Marine Fisheries and Oceanology. We have the links sent on the chat box. If you want to apply, we are currently accepting applications for the Master of Science in Fisheries Biology and Master of Science in Ocean Sciences. Please go find the link on the chat box because there you can find the details to apply for scholarships and also to the program. Thank you so much for attending our webinar. We wish to see you again on August 8th at 2 p.m. for the talk of Dr. Chihao She. And the evaluation form, I think it's also on the chat box. We will also post the evaluation link on the FB Live post. So you may also get the evaluation form there so you can get your e-certificate. Thank you so much. We have a total of, we reached about 360 people on Facebook and then 260 here on Zoom. So it's very interesting that a lot of people are already starting to love working on zooplankton. Thank you so much. See you again. Sir Juni, we can end the Facebook Live.
Okay, thank you. Thank you for your time.
Thank you so much. Dr. Machida, thank you very much for doing this.
Yeah.
Please do introduce us to other scientists that we can invite for the lecture series. Thank you,
Thank you.
Director Sheila, and the committee for the webinar.
Thank you po sa mga UPV
Bye.
people.
Louie, go to sleep na. It's very late.
Yeah.